Good day everyone, I am Emeline B. Casas from BSED 3A. Today, I am going to discuss about Enterococcus faecium. What is Enterococcus faecium? Enterococcus faecium is a gram-positive, gamma-hemolytic or non-hemolytic bacterium in the genus Enterococcus. It can be commensal innocuous coexisting organism in the gastrointestinal tract of humans and animals, but it may also be pathogenic, causing diseases such as neonatal meningitis or endocarditis. Generally, a faecium is an ubiquitous commensal microorganism commonly found in the gut of animals and humans. Let's proceed to the characteristics of Enterococcus faecium. It has an average size of 1 to 2 mm. Its shape is a gram-positive, cocal-shaped, facultative anaerobic bacterium that can occur in pairs or chains. Aphysium arranged individually in pairs or short chains. Optimal temperature for growth of Aphecalis and Aphysium is 35 degrees Celsius. Aphecalis and Aphysium are normal inhabitants of the intestinal tract, female genital tract, and less commonly oral cavity. Its unique cellular structures is gram-positive cells have a thick peptidoglycan layer along with teichoic and lipoteichoic acids. It has circular DNA as well as several plasmids. It is capable of conjugation through the release of sex pheromones and secretes of aggregation substances and also forms biofilms. The cell has pillae and flagella. Let's proceed to the nutrition. One of the major benefits of Aphysium is that it is uniquely suited to survive the digestive process and flourish in the gut. It promotes a balanced gut environment by competing for resources that harmful organisms would otherwise consume and use to grow. Next is the supplementation of food with Enterococcus faecium stimulates immune functions in young dogs. Puppies were allotted to two groups, receiving either a controlled diet or a diet supplemented with 5 times 10 colony forming units of probiotic Enterococcus faecium from weaning to 1 year of age. Fecal and blood samples were collected from the dogs at different time points for the measurement of fecal immunoglobulin and the proportions of lymphoid cell subsets. Let's proceed to the pathogenesis and control of Aphysium. Aphysium is a human pathogen that causes urinary tract infections, endocarditis, surgical wound infections, and nosocomial bacteremia. Nosocomial infections occur in a hospital setting during the treatment of a prior illness. Aphysium contains a number of virulence factors that increase its degree of pathogenicity. Enterococcal surface proteins are used by the microbe to aggregate and form biofilms. Aggregation substance allows the microbe to facilitate the transfer of genetic material between cells by targeting them. A protein called cytosolin is found within the microbe's cytosol and is responsible for the lysis of erythrocytes. Its control is linezolid, an oxazolidinone antibiotic, is available orally and intravenously and is used to treat infections caused by Aphysium and Aphecalis strains, including vancomycin-resistant Enterococci. Before treatment of enterococcal infections, all suspected intravenous lines, intra-arterial catheters, and urinary catheters should be removed, if possible, and abscesses drained. Infections that do not require bactericidal therapy are usually treated with a single antibiotic directed toward Enterococci. These infections include UTIs, most intra-abdominal infections, and uncomplicated wound infections. Medical or economic importance of Enterococcus faecium. Some A. faecium strains are used as probiotics and are ingested in high numbers, generally in the form of pharmaceutical preparations. Such probiotics are administered to treat diarrhea, antibiotic-associated diarrhea, or irritable bowel syndrome, to lower cholesterol levels, or to improve host immunity. In animals, enterococcal probiotics are mainly used to treat or prevent diarrhea, for immune stimulation, or to improve growth. It is used as an alternative to antibiotics in the livestock industry. Enterococcus faecium acquired traits that allowed it to become a successful nosocomial pathogen. A faecium inherent tenacity to build resistance to antibiotics and environmental stressors that allows the species to thrive in hospital environments. Let's proceed to the impact of Enterococcus faecium into life. Some strains of A. faecium may be pathogenic and harmful to humans and can cause bacteremia, endocarditis, urinary tract, and other infections. Moreover, antimicrobial resistance to many commonly used antibiotics has been reported, and A. faecium is the leading cause of multi-drug resistant enterococcal infections in humans. It is thought that about 70% of A. faecium strains in US hospitals are resistant to vancomycin and 90% are resistant to ampicillin. A. faecium resistance is acquired by gene transfer systems such as conjugative and non-conjugative plasmids or transposons. That's all. Thank you.